So, I'm officially changing name, right?
Oh, no. It's like, because of the picture, I think it's supposed to say Sanford. I was trying to figure out how to, what it's supposed to
Is
say.
that really? Is that some guy that looks like
You had to wear a lab coat to wear a white.
Yeah.
It's a little house with a little door. That's so weird.
It's just gonna not follow me. Like, it's just gonna be off.
Just don't go quite so far. Can't run into the corner anyway.
Is it forward?
It's not on the screen, but I'll be fine.
If you're ready, you can go ahead and start, Carly.
Yep.
Take one step.
Hi, I'm Carly Sparks. I'm a senior at Armour High School, and for my senior project, I chose to make a DNA protein, to like genetically modify it to make a new one. I chose this project because of my 3 grandfathers. This is my grandpa, Dove. He had cancer. My grandpa Dennis had cancer, and my grandpa Jerry had cancer. These 2 of my grandfathers both died of cancer. So that's kind of where my cancer researching kind of started. I kind of got inspired by them. And then I also really have a love for science. I'm kind of a science nerd, so this project fits right along with that. So my research paper was trying to find out like the health effects of secondhand smoke on the human body. I kind of chose this because my grandpa Dennis, he died from smoking of lung cancer, so this kind of fit in very well with that. And my thesis was that first and secondhand cigarette smoke causes lung cancer, and therefore the United States government should ban all cigarette smoking. Through the research of this, I actually found out that first-hand smoke has harmful effects, as everyone probably knows, but secondhand smoke has the same effects or even worse effects at time, which I didn't know. So what does my project mean? It's kind of confusing to everyone probably here. So there is a protein, and a protein is something inside of your body that helps with your bodily function. So when you blink your eye or when you use the restroom, proteins are helping with that. So there's a new protein, not really new, but a newly discovered protein that Stanford research has found that has been known to cause cancer when it's overworked. There's a part of this protein, a little tiny part of this protein, called called the PZ domain. So a little piece of this protein called the PZ domain is what causes the cancer when it's overexpressed. And so my ultimate goal for this project was to create a new protein. So I was going to take this FNB1 protein and take out the little chunk of the PZ domain to see if it could function still without causing cancer. And then the new protein that I made would be sent on to the Vermeer lab at Stanford Research for further cancer research. So a quick run through of the process that I went through. So in the first step that I did, it's called quick change mutagenesis. So what I did in this step is I designed a primer. So a primer is something that unzips a cell. Basically what it does is like a coat or zipper on your coat. It'll unzip and then it zips itself back up. The purpose of this was to get the chunk of the FMB1 protein called the PZ domain out so that the protein can still function. The primer also helps to break down the cell wall. So it's kind of like when you eat something, your food is breaking down in your mouth. It's kind of the same concept. In the next step, DNA transformation, the DNA that I used, it was prepped to be injected into the cell. So the primers were used and other different chemicals for this step. And then I also took DNA, human DNA, and injected it into the cell. The next step was extracting and purifying my DNA. Basically what I did in this step is after the bacteria cell with the DNA grew on a plate like this, all these little specks are called colonies of DNA. So all of these little specks have cells of bacteria with DNA in them. This is what it would kind of look like under the microscope. So all these little circles are cells. So what I did is, if it looked healthy, kind of like this one right here, or this one looks better. So if it kind of looked like that, or looked bigger from here, I took it, I plucked it, and then I cleaned it using different chemicals to purify it. So my next step was to analyze the DNA, and what I did in this step was I used this machine right here called an epoch machine. So what this machine does is this right here, it opens up and it allows me to put a very, very small part of my sample in there, and it's going to tell me how much DNA is actually in that sample. And then the last step that I did was sending it off for sequencing. Stanford Research, which is where I worked for this project, they don't actually do the sequencing themselves, so it has to get sent off. What sequencing is going to tell you is it's going to tell you the DNA base pairs of the DNA samples. So what this means is that your DNA inside your body is made up of different chemicals. The abbreviations for these chemicals are A, C, T, and G. So that's what makes up the DNA in your body, and that's what this was telling me was the sequence of all those. So it would tell me if there's C, G, T, A, or vice versa. So it's going to tell me what the DNA is made of, what the order of the chemicals is for that. So the next step for my project after it was all done was to make my poster, and I'll get into that a little bit later. And then I went to the Regional Science Fair in Mitchell, and I ended up winning a couple awards there from my work. I got first place in the Senior Life Division. I got a Naval Research Award, a Thielen Research Award, a Scholarship Award to DWU, and another Life Science Award. So this is probably really confusing, and it's still really confusing to me with all the abbreviations, and I don't even know all of them. So basically what this is it's a bunch of different proteins that Stanford uses, or that is being used for cancer researching right now. So the protein that I worked with, FMB1, is right about here. These numbers right here, they stand for how many base pairs make up the protein. So mine was right here. It had 5,280 different base pairs, so it's located right about here. So this is, like I said, is just a bunch of different proteins and stuff that Stanford uses for their research right now. So the results of my project. So the last step of my project in order to get these results was the sequencing. So what the sequencing would show me, like I said, was the base pairs of my DNA. The program that was used, that the company that I sent them off, what it told me was that my results for this project were inconclusive. So what that means is that the program couldn't tell what the DNA base pairs were. It knew what some of them were, but it couldn't tell me definitively what they were. And so that also means that I can't tell if I successfully did this project or not. I had other samples sent off, but like I'll explain later, it takes a tremendous amount of time to wait for these sample results to come back. And then, so right here, this is also probably confusing, but all these spikes, they indicate what the DNA base pair is. So as I said earlier, T, T, G, all of these up here are the chemicals that make up your DNA. The spikes will determine what chemical is there. So this is what sequencing results should look like. So after I got them back from the company, this is what they should have looked like. And this is what they looked like. So it's really small to read, but in order for me to get a full page, this is what it has to be like. So there's 3 pages of this, because as I said earlier, there's supposed to be 5,280 letters, and there's none. So this means, like I said before, that it was inconclusive, and I'm waiting on results still right now. But you can see that these dashes, it doesn't know what it is. It has a little bit of indication here, but it still can't determine. And then finally, it determined right here that there's an A. And then all the way down here, there's a couple. But basically, this is what all my results look like. All 8 samples were like this. This is probably the worst of it, but this is what they look like. So to see if I successfully did this project, I had to do an alignment with an alignment program that Stanford uses. So right here is the DNA base pairs of the FMB1 protein. Right here is that vector map with all those other proteins that I showed earlier. So there's the base pairs of that. Right here is the little chunk, PDZ domain. That's the little chunk that I was supposed to take out of 5,280 of these letters. That's what I was supposed to take out. And it was a very difficult process to try to do that, and I'm still waiting to see if I did do that or not. But as of right now, I don't know if I did do that or not. So these are my DNA concentration numbers. So basically, these are telling me how much DNA is in my sample. So this is the name of my sample, and then the DNA concentration. This stands for mouse FNB1 delta PDZ. So FNB1 is the protein. Mouse is because some of them were mouse. And then the delta PDZ means without PDZ, because it was supposed to have no PDZ domain. And then, so these numbers showed me that there was DNA in the samples before I sent them off, which is a good thing, and that's what I wanted. It doesn't really matter how much DNA was in there, because like, as you can see, like this one is 99 and this one is 35. So it doesn't really matter how much DNA is in there, it just matters that there was DNA in there. So the stretch for my project was that I've never done procedures like this before. I've worked with Stanford, this is my 3rd year working with them now, and this is the most complex project out of them all that I've done. I worked on my people skills a lot, because I'm not very good with meeting new people and interacting with people, and so that was kind of difficult for me to do. And I've never worked in an actual lab. I mean, the lab we have at the high school is a lab, but it's not as great of a lab as Stanford is. And I worked on my scientific writing skills because, as I'll explain when I get to my poster, it's hard to write like summaries of what I did. So the problems that I encountered were getting results back. My mentor, she told me that my results will be back within 5 days, and it took 2 months. So I was supposed to be done with my project in January, and I didn't get done until March. I got done a week before the science fair. So that was interesting to try to get my poster done and cram all that in. And then the procedures, like I said before, they were really difficult to follow. They were really precise, and if you messed up, it could mess up the whole experiment. And communication was really hard too, because with my mentor being in Sioux Falls, and she runs the Promise Lab up there, so she's very busy, and it was kind of hard to communicate back and forth with her. This is my budget page. So it might scare some people that this number right here is $9,000. That's only because the equipment that I use is worth $9,000. That's how awesome Stanford is that they let me use the equipment for nothing. So I actually only spent about $97, which was great. I overestimated by $9,048. So I learned that, like I said before, that the PDZ domain, which is a little chunk of that protein, it can cause cancer if it's overworked 90% of the time. I also learned that first and second-hand smoke can both have a very harmful effect on your body, and that complex procedures had to be taken one step at a time, because if they're not, And if you're like me and you just want to rush into things, it just doesn't work out. And believe it or not, a genetically modified protein is very hard to make. And I also learned that science doesn't work out the way you want it the first time. That's very hard for me to deal with because I like things to work out the correct way the first time. I learned that I have to have a little bit more patience. I'm sure the Schneiders can testify that I don't have very much patience when it comes to projects like these. And my confidence was really tested this year because in the past years at the science fair, I never really went into, like, boosted myself up for what I did and not what I was supposed to do. So that was really new for me this year, and the reason I won a lot of awards there is because I really boosted myself up and said, this is what I did. This is, you know, even though I failed, this is what I actually did. My learning ability is a lot greater than I thought it was. This project that I did is, and my mentor said so too, is that it's a graduate level project. And so my learning ability was really tested this year too. And I also learned to stay calm. 5 days, and it took 2 months, so my patience and staying calm was really tested. My project affects my future plans because I plan on either working at Sanford Research as a cancer researcher or teaching, and this project allows me to explore both areas by researching and teaching my project to many people. I can use this later in life for internships and researching if I decide to go into researching, or teaching if I do that. So now my poster.
Turn the light on up here.
So this is my poster that I used to enter the science fair. Most people probably have kids that did the science fair in middle school, and they had those foam boards or the cardboard boards. And for me, going into high school and the science fair is really, really, really competitive. If you think that I might be a nerd for science, you should see some of the other people there. They're like lunatics when it comes to this stuff. But, so, I said my scientific writing skills were tested a little bit because for all these steps, what I explained to you, I have to explain that to judges too. And I can't put everything that I did on my poster because, one, there's not enough room, and two, they don't like if you put every little detail that you did on your board. They want you to explain it. And so I really had to summarize everything I did, like over here in my discussion and over here for my steps and my introduction and stuff. So that was something that was really difficult for me to do too, is to summarize. And this took a very long time to do. Probably 3/4 of the time I spent on this project went to this, because it's very important that you present it well and that you do it well for the judges at the science fair. I want to say thank you to my mentor, Liz McMillan at Sanford Promise. She was a really big help with this project. I'd also like to thank my advisors, Mr. Barker, Mrs. Bitterman, and Mrs. Dockweiler for keeping on me, especially when the science fair deadline kind of got close. And for Mrs. Dockweiler for listening to me complain and giving me class time to present my project. I'd like to thank Mrs. Ziegler because I probably wouldn't have gotten my research paper done like many people if she wasn't there. And the Schneiders for also listening to me complain and helping me with my poster and being here in Armour instead of being up in Sioux Falls. I'd like to thank Paul Colbert and Pay Vermeer at Sanford Research. That's the lab I work for, the Vermeer lab. My parents for helping to finance this project. Mr. Cram for letting me do this project, and the judges and audience for coming to listen to me today.
Couple of things first. One, they all say they want to thank Mr. Perrin for letting them do the project. So what you're gathering from that is these kids come to me in October and says, please let me do a senior project, Mr. Perrin. And I don't remember any of them begging me to let them do senior projects. So, but anyway, thank you. Secondly, tough thing to do because this audience is
That science-
way below what you're talking about, girl, so, I mean, it's tough to present
Yeah,
that stuff, I mean, that, you know, I don't know what else you do, you did a good job with it, but I mean, I hope there's not a quiz. And then third, just point out, you talked about boosting yourself up, and then you said, even though I failed, get that out of your vocabulary, right? All of you, you didn't fail, just because things didn't come up, that's science research, as has been told. So just that phrase, you know, that phrasing as you go forward, because you're probably gonna do a lot of this stuff, don't use that term fail, because I don't think anything you did here, you know, I see science people saying that,
Not even
you know, I mean, yeah, you bet, brag yourself up. Failure is a bad word for you,
Your
for
result-
any of these people to use.
Your results were negative.
yeah.
Okay, now I'll be quiet.
Good, good.
Yeah, we don't have any questions.
I wanted to go back.
How do you weld?
I want to go back, I want to contradict some of this.
Yeah, that,
Really?
I'm not even sure I myself could do that.
so,
Good job.
so do you plan on following up on this project?
If the results come back. And my mentor is really busy this time of year because she has a lot of, like, high school groups and, elementary groups that come into their lab. So it just depends on.
But I
when she
mean, it
gets
may be in
back.
college if you have to do a project,
Yeah.
because I'm sure it's till you cure cancer.
Is Dr. Vermeer doing similar experiments?
Yeah, she's doing the same thing right now. So basically what I've done the last 3 years is kind of just follow what she's doing in her lab. So just done projects on what she's kind of doing in her lab. That way she's doing the same thing and I'm doing the same thing. So if I have questions, she knows what she's, well, obviously she knows what she's doing, but she's working on the same thing. So she has easy access to if I need help, so.
Do you know why it took 2 months instead of 5 days?
Yeah, because there was a lady that worked at Sanford that was getting the results, but she doesn't work there no more. And so they were getting sent to the wrong person, and it took 2 months to get it figured out. So, yeah. But it all worked out.
Good job.
Thank you.